Yes, boys. Today we will talk about the GIT start. I will tell you the slides of GIT in two classes. As you have read in your previous classes, GIT starts from oral cavity and continued up to the anal orifice. This whole part of GIT, the inner surface of this whole part of GIT is lined by mucous membrane. That mucous membrane varies from different parts of the body. In oral cavity, the slides which you will see that will talk. Tongue is a muscular organ, and this muscular organ is consisting of skeletal muscle fibers. Underline that skeletal muscle fibers. No smooth muscle, only the skeletal muscle fibers bundle is forming the tongue. Both the surface of tongue, that is dorsal surface facing upwards, visceral surface facing downward. Both the surfaces of the tongue are lined by the mucous membrane. That means on the dorsal surface as well as on the visceral surface. But we find that the mucous membrane lining on the dorsal surface of the tongue don't tell me repeatedly that I should make you out. Why don't you sit properly? And you boy also, not you. If you are not serious in the class, why are you coming? Just for the sake of attendance? Don't come in my class for attendance. I never took the attendance. I completely live on yourself, whether you want to become a good doctor or not. I least bother about the attendance. And please don't disturb me. The dorsal surface of the tongue is rough, while the visceral surface of the tongue is smooth. The reason why it is rough, the answer is that the dorsal surface of the tongue is lined by large number of papillae. Papillae are the projections of the mucous membrane. So the mucous membrane of dorsal surface of the tongue is showing the large number of the projections, and these are called the papillae. And these lingual papillae are very much present on the dorsal surface of the tongue, but they are not present on the visceral surface of the tongue. So one is the lingual papillae. Another is lingual tonsil. Both lingual papillae as well as lingual tonsil, they are present over the part of dorsal surface of the tongue. They are not present on the visceral surface of the tongue. Besides this, if we see the dorsal surface of the tongue, the dorsal surface of the tongue is divided into two parts, anterior 2/3 part and posterior 1/3 part. See this diagram. This is the anterior 2/3 part and this is the posterior 1/3 part. In the anterior 2/3 part, the mucous membrane is showing the large number of the projections, and these are called the papillae. So papillae are present on the maximum part on the anterior 2/3 part of the tongue. If we see the diagram, you can see that it is dividing into two parts by the V-shaped line. This is V-shaped line, and this V-shaped line is called sulcus terminalis. So sulcus terminalis is dividing the dorsal surface of the tongue into two parts, anterior 2/3 part and posterior 1/3 part. In anterior 2/3 part, the large number of the papillae are present, while in posterior 1/3 part, you will find just a lymphoid tissues, and these are called lingual tonsils. Besides this, one more thing. When we talk the different type of papillae, the one type of papillae which is rich in along the V-shaped sulcus, I am talking of this. So the papillae which are present just anterior to the sulcus terminalis are very important because they are very rich in the taste buds, and these are called circumvallate papillae. So circumvallate papillae are very much in enormous amount present along the sulcus terminalis. No more latecomers or still? For the latecomers, I am just telling you, hurry up. Why don't you take a breakfast? The tongue is muscular organ, and it is muscles are only the skeletal muscles, nothing else. Never say the smooth muscle. It is a muscular organ of the skeletal muscle which is covered by mucous membrane on dorsal surface and visual surface. The mucous membrane on the dorsal surface of the tongue is giving the projections which are called the papillae. Papillae are of different types. The dorsal surface of the tongue is dividing into the anterior 2/3 part and posterior 1/3 part by V-shaped sulcus, that is sulcus terminalis. After this, I have to stop for the latecomers. गेट बंद कर दो. All right. After seeing its dorsal surface and ventral surface, when we see the transverse section of the tongue in histology, we will find that tongue is following the same pattern of the GIT. That means the epithelium is lined by stratified squamous non-keratinoids. But one thing is very important here, no doubt. Maximum part of the dorsal surface of the tongue and ventral surface of the tongue, it is lined by stratified squamous non-keratinoid epithelium. But at one type of papillae, which is cone shaped, look here, that we call the filiform papillae. At the tip of filiform papillae, it is slightly keratinized. Okay, so when the students replying very well, then we asked, all right, tell us, is it stratified squamous non-keratinized completely? To get good marks, you should reply that, sir, at the tip of filiform papillae, it is showing the keratinization. Otherwise, rest of the part, it is completely stratified squamous non-keratinized. Why it is keratinized at tip of filiform papillae? Because whenever we eat the food, no necessity in the smooth food, but when you are eating the non-veg or whatever the hard food, by that time the filiform papillae is working more, and as it is working more, filiform papillae during the hard food, so naturally there will be the epithelium will be ruptured. So for that, it is at the tip of filiform papillae there is keratinization to protect the epithelium. Okay, all right. Besides this, the epithelium is showing the large number of the papillae. What are the papillae? Papillae are the projections of the epithelium. And below the papillae, there is the part lamina propria. This whole is the lamina propria. Lamina propria is a connective tissue. Lamina propria is connective tissue which can contain lymph nodes, blood vessels, like that. Depending upon the section, The lamina propria may contain the lymphatic tissue or the nervous part. All right. As we go down, look this part. There are the two type of pictures in the diagram. One is the longitudinal section, another is transverse section. These are the muscle fibers, skeletal muscle fibers, all type of the skeletal muscle fibers. They may be seen longitudinally cut, they may be seen transversely cut, or they may be seen oblique cut. So all these are the muscle fibers. And what type of muscle fibers? Skeletal muscle fibers. The section may be oblique, maybe the longitudinal. Here it is shown the longitudinal section, transverse section, and all these. Besides this, you will also find there the glands. The glands may be the serous gland or the mucous glands. All these structures will be seen in the tongue. All right, we identify that the section is tongue. Next point, we want to confirm ourselves that what type of papillae are seen. In this diagram, the two type of papillae are visible. This is the filiform papillae. Filiform papillae are conical shape, taller in length. Epithelium is same, only the difference is in the shape. Filiform papillae are conical, like this. And between this filiform papillae, you are seeing this type of papillae. This papillae is comparatively wider more than the filiform papillae, and its surface is also wider. Or you can say that this is mushroom shape. And this type of papillae is called the fungiform papillae. F-U-N-G-I, fungiform papillae. Look the difference between the fungiform and filiform. In filiform papillae, there is no such picture which is shown here in fungiform. That is the taste bud. So taste buds are present in fungiform papillae. No taste buds seen over the filiform papillae. Filiform papillae is without taste bud. Whatever the food we are eating, the taste we are feeling, that is taken by the taste buds. And these taste buds are very much in number over the circumvallate papillae, which I will show you now. So taste buds are present at the two sides. One is fungiform papillae and another is circumvallate papillae. Now see the another. Now look this one. This is circumvallate papillae. The circumvallate papillae is more larger, and if you convert the cone, flask, just like a flask shape, surface is more wider and it is very rich in their taste buds. Another thing, see on both the sides, you can see the groove are present on both the sides of circumvallate papillae. So this is the circumvallate papillae. So identification of the tongue is very simple. Okay, and you can also identify the filiform papillae, fungiform papillae, and circumvallate papillae. Filiform papillae are enormous. Circumvallate papillae are very much in number at the V-shaped sulcus of the tongue, and can also see the fungiform. Besides this, sometimes the question asked, maybe in the MCQ or in short notes, what are the taste buds? What are the different type of the cells present in taste buds? Look this diagram. Before coming to the taste buds, see the different type of papillae. This is the filiform papillae. Again, this is the fungiform, and this is the circumvallate papillae. And in the papillae, what is the epithelium lining the papillae? That is stratified squamous non-keratinized. And between these cells of epithelium, you will find these structures. All these are the taste buds. And similarly, here in the fungiform, taste buds. But no taste buds shown over the filiform papillae. Now look, this is the diagram of the test words. So how we identify the test words? Between the epithelial cells, you will find the flattened cells. Flattened cells. Look here. All this is the epithelium. This is the epithelium. Between the epithelium, this slightly elongated cells. These are the test words. From the next diagram, see. These are the different cells present in the test words. Look here. This is the receptor cells. The receptor cells at its one end is opening in this open space. That is called test pore. So one end of the cells, whatever the cells, whether it is the gustatory cells or test cells, supporting cells, or besides the third cell, which is the visual cells, I am showing you what are these different. Look this one. This is the part receptor cells. So one end of it is opening here in this cavity, that is oral pore, and another end is downward. At the other end of the cells, we attach the nerve fibers. Okay. Now, what happens? As the food is, after converting into the chyme and this, as the food comes in contact with the parts, it receives the sensation, and that sensation through the nerves reaching to the brain. There it has been analyzed that what type of food, whether the sweet, sour, all these tastes are then selected by the brain. Only the taste buds, they carry the sensation from taste pore through the nerve fibers to this. Another this part, the second cell is the supporting cells, which is also named as sustentacular cells. Their function is they provide the support to the gustatory cells. And third type of the cells, madam, please go. What is the time? You are coming late half an hour. The third type of cell is the visual cells. So these are the three types of cells present in taste buds. After the tongue, we come to the next slide. That is the part of esophagus. The esophagus is about 25 to 30 millimeter tube, which is connecting above with the pharynx and downward with the stomach. This muscular tube is lined by, again, the stratified squamous non-keratinized epithelium. All right. It is having the lamina propria, that is this part, and this lamina propria, and below that the part musculus mucosa and then musculus externa. So before talking of this part, I am telling you the general plan of the GIT. Whenever we see the slide of GIT, what are the structures we will come across? First one is mucosa. We use the word mucosa, that means it is including these three parts. Mucosa including epithelium, first. Second, lamina propria. Note कर लो, तुम्हारे से जरूर पूछूंगा. Second is lamina propria, third is muscularis mucosa. All these three structures combinedly forming the mucosa. So whatever the slide of GIT we will see that show these pictures. One is mucosa. Below the mucosa, there is part of submucosa. And third, muscularis externa. And outermost is serosa or adventitia. These are the basic things we will see in the GIT slides. Only the variations come at different parts of the GIT. When we see the slide of stomach, we find there is slightly variations come, like small intestine, jejunum, ileum, appendix in everywhere. But in every slide we will see like this. Mucosa, in mucosa first thing is epithelium. Look here, the epithelium is this one. The epithelium, first of all, after looking the epithelium, confirm whether it is the stratified squamous non-keratinized epithelium or simple columnar epithelium or ciliated columnar epithelium like this. So this is the stratified squamous non-keratinized epithelium. All right. Second thing, it is showing the projections and invaginations. The projection part is slightly raising above, and invagination is this part. Below the epithelium, we find the lamina propria. You must be clear about the lamina propria is a connective tissue part. It is a connective tissue part, and connective tissue part may have the glands or may have the nerve fibers like that, depending upon the part, which part we are studying. See here, in this lamina propria, slightly glands. Besides the gland, the part is also showing the nerve fibers and slightly lymphatic tissue. The third part of the mucosa, that is muscularis mucosae. In case of esophagus, the muscularis mucosae is consisting of single layer of the elongated smooth muscle fibers. I am using the word smooth muscle fibers, underline. In all the part of GIT, you will find the smooth muscle fiber maximum, except the few places. All right. This muscularis mucosae is consisting of circular smooth muscle fibers. In most of the part of GIT, the muscularis mucosae is consisting of two layers. Inner is circular smooth muscle fiber, outer is longitudinal muscle fiber. Most of the part, except this esophagus, where we find only the single layer. One thing have you note? Note down this part. What is this part? This is the part of lamina propria. In the lamina propria, you are seeing the large number of the glands. Large number of the glands. And these glands are mucus secreting glands. And they are present in the esophagus. So they are also named as esophageal glands. So keep one thing in your mind, esophageal glands are the mucus secreting glands. And they are present in lower part of the lamina propria and also in the part of submucosa, extending into the submucosa. Next to this, then we come to the part of muscularis externa. Muscularis externa is again the two layers of, before that, see the part, the submucosa is not so clearly shown, only the glands are visible. The thing which I want to tell you, one which is not shown here, that is the nerve plexus. In submucosal parts of esophagus, there is the nerve plexus of parasympathetic fibers that is called Meissner's nerve plexus. Meissner's, M-E-I-S, M-E-I-S-N-N-E-R-S, Meissner's nerve plexus, which is seen in the part of submucosa. So submucosa is showing the part of the esophageal glands which are very much seen in the part of the muscularis mucosa just above the lamina propria and also in the submucosa. Besides the mucus, the esophageal glands, in submucosa there is also present nerve plexus which is commonly asked. Besides the esophageal glands, what else you find there? That is Meissner's nerve plexus. And what is the Meissner's nerve plexus? These are the parasympathetic fibers which are supplying to the part of the smooth muscles and the part of the glands in the esophagus. Next to this, you will find the bundle of the skeletal muscle fibers. Look here, this is the external muscularis cord, and this muscularis external cord is consisting of two layers of the muscle fibers, inner circular, outer longitudinal. And one thing, I am using the word circular. Look how the circular muscle fibers seen. And elongated muscle fibers are seen in these small bundles. Why? Longitudinal to longitudinal दिखना चाहिए. Look, this is the section we are taking. Circular muscle fibers are like this. So how we take the section? Transverse section like this. So circular muscle fibers are seen like this, and elongated muscle fibers are these. So longitudinal layer of the muscle fibers and circular muscle fibers. So inner layer is consisting of circular muscle fibers and outer is longitudinal muscle fibers. Outermost is adventitia, that is consisting of loose connective tissue, nothing else. In muscularis external coat, that is inner circular and outer longitudinal, there is also present the nerve plexus, and that nerve plexus is called myenteric nerve plexus or Arbeck's nerve plexus, that you will find in all GIT parts. In all GIT parts, submucosa is showing the Meissner's nerve plexus, and muscularis externa, between the two layers of muscularis externa, there is present the Arbeck's nerve plexus or the myenteric nerve plexus. All right. Besides this, the next question of the examiner, you have identified that slide is of esophagus, replied well each part. But next question will be whether the section of esophagus is from upper 1/3 part, middle part, or lower 1/3 part. For the confirmation that section is from upper 1/3 part, middle 2/3 part, middle part, or lower 1/3 part, we see only the muscularis externa. All the part is same, epithelium is same, lamina propria is seen in all the three parts, whether the section is upper 1/3, middle part, or lower 1/3 part. All these three are showing the epithelium, lamina propria, the esophageal glands, muscularis externa, each and everything. For the differentiation of the upper 1/3, middle 1/3, see only the muscularis coat. The muscle coat, that is inner and outer, in upper 1/3 part is purely consisting of skeletal muscle fibers. Underline that. That is very commonly asked in MCQ and also Viva. If the section of esophagus is from upper 1/3 part, the muscularis externa is consisting of only skeletal muscle fibers. If the section is taken from middle part, the muscularis coat is consisting of both type of the fibers, skeletal as well as smooth. If the section is taken from lower 1/3 part of the esophagus, the muscularis externa is consisting of only smooth muscle fibers, nothing else. That is very important, commonly asked in the MCQ or maybe BIBOC. So for the differentiation of the esophagus, and once you say the slide is of esophagus, next question will be this. Section is of upper 1/3 part, middle part, or lower 1/3 part. Your reply will be in upper 1/3 part, it is showing only the skeletal muscle fibers. Where? In muscularis mucosa? No. Only in the muscularis externa coat. Okay, clear? After this, I come to the next slide. Stomach. Esophagus is continued into stomach. So, for the stomach, again, we will see the epithelium, that is mucosal part, submucosal part, muscularis externa, and adventitia. In mucosal parts, the epithelium of the stomach is lined by, see the diagram and speak, I will not tell. Simple columnar. Epithelium of stomach is simple columnar. And this simple columnar epithelium is showing the invagination. Look, this is the invagination, this is evagination. This is evagination, and this going inwards, that is invagination. So the epithelium which is going down, invaginate, that is called gastric pits. So these are the gastric pits, and these are the projections are called rugae. I use the word rugae because when the stomach is empty, the rugaes can be seen. But stomach when full of food, the rugaes disappear or pressed. So the epithelium is showing the evagination, that is this one, and also showing the gastric pits. In the slide of stomach, there is two part of section. One is fundus part, another is pyloric part. So first of all, you confirm whether the slide is of stomach or not. After confirming the slide is stomach, next is whether it is fundus part or pyloric part. So these two things we will see in the slide of stomach. First, how we confirm? Look the epithelium. You are finding the simple columnar epithelium. And as we go down in the gastric pits, in the gastric pits are consisting of, between the epithelial cells, you are seeing some cells are acidophilic, red color or acidophilic. And between the acidophilic cells, some cells are basophilic or blue color. Have you noticed down, note down this thing? This simple columnar epithelium of gastric pits is showing two types of cells. Some cells are acidophilic or red color. Between the red color cells, some cells are basophilic, blue color. The cells of blue color cells or basophilic cells, these are called the cheap cells. And the red color cells, these are called the parietal cells. The parietal cells, they are secreting the acid. Parietal cells are secreting the acid, and that acid acting over the food and converting this food into the chyme. Chyme is a bulky pulp mass of the food. So the action of the other enzymes can take place. We eat the food, maybe the bulky or whatever the type of the food. That food, first of all, acting by the parietal cells of the stomach. The parietal cells of the stomach converting the food into the chyme. Chyme is a smooth mass or pulp. Over that pulp, the another enzymes or hormones, they will act. So that is the action of the parietal cells. Parietal cells are very larger in number in the fundus part of the stomach. See this diagram. आपको रेड कलर के सेल्स ज्यादा दिख रहे? Yes or no? I will send all of you to the ophthalmology department for checking of your eyesight. रेड कलर के सेल्स दिख रहे नहीं दिख रहे ज्यादा? So parietal cells are more in number in the part of fundus part, and these are the transversely cut section of the gastric pits. Rounded structures are transversely cut section of the gastric pits. And between the parietal cells, very less number of the basophilic cells or the cheap cells. Okay, so fundus part of the stomach we identify. And below this, epithelium and lamina propria. What is this part? This is the part of the rugae of the stomach, and in the rugae of the stomach and in the gastric pits, you are seeing this whitish color part. That is the lamina propria. That is, this whole part is the lamina propria. Lying in the lamina propria, you can see the blood vessels, and also depending upon the part, here the lymph tissue are not so much in the stomach. And lower down, this is the part of muscularis mucosa. And next to this is the part of submucosa. Then we come to the part of muscle coat, inner circular and outer longitudinal. So the other picture is the important part. See the epithelium. In the epithelium, see the gastric pits, and in the gastric pits and in epithelium, another thing you will note down, that is parietal cells and the cheek cells. Okay. The number of parietal cells you will find more. You can easily say that the section is of fundus part of the stomach. Next to this, see this diagram. Again, the epithelium is simple columnar epithelium, no problem. And gastric pits you can see also. And below this, the gastric glands also seen. So, this is the section of, again, how we will identify whether the slide section is of fundus part or stomach. First of all, whether the section is stomach or not. For identification of the stomach, see the parietal cells and cheek cells. That is very important. Without seeing the parietal cells and cheek cells, don't commit that the section is of stomach. Okay. After identifying in the pyloric part, the cheek cells are more in number. They are seen basophilic. Cheek cells are more in number. The parietal cells are few. Their number is reduced, but not absent. Mind it. In pyloric part, the parietal cells are few, but the cheek cells are more. And second point, see the gastric pits. Gastric pits are more deeper. Gastric pits are more deeper in pyloric part of the stomach. So by these important points, first is the cheek cells are more in number, parietal cells are few, and gastric pits are more deeper. So section is of the pyloric part of stomach. Then I come to the next slide of the GI. When we come to the part of small intestine or large intestine, for identification of the or the differentiation of the slide, in the case of small intestine, which is consisting of duodenum, jejunum and ileum. In the large intestine, the transverse colon, ascending colon, transverse colon, descending colon, rectum and anal canal. First of all, in a small intestine, when we see the slide, we find the villi in case of jejunum, ileum. That means in a small intestine, you will find these villi. Villi are very minor projections of the mucous membrane, which are seen in low microscope or in the light microscope. They are seen just pink color line in hemotoxin eosine. Lying over the epithelium, whenever we see the slide of GIT, of small intestine, what we will find that lying over the columnar epithelial cells, we will find the pink color area. That pink color area is of microvilli. But that pink color area, when we see under the electron microscope, it is showing the minute projections or very minute projections. The purpose of the villi is to increase the surface area, nothing else. Because as the food from the stomach and after the duodenum comes into the jejunum and ileum, the food remains in longer contact with the part of the GIT, so the absorption of the food can be easily taken place. That is the reason why the villi are present in the small intestine. Another thing, between the columnar cells, there is present the goblet cells. Between the columnar cells, there is present the goblet cells are mucus secreting cells. The important part, as we start from duodenum, the appearance of the goblet cell starts. Look the diagram of the stomach, esophagus. I have not talked of the goblet cell. No goblet cell is present in the epithelium of the esophagus, nor in the tongue, nor in the part of stomach. the goblet cells make their appearance from the duodenum. As we go down, the number of the goblet cells are increasing. In the large intestine, the number of the goblet cells are more than the columnar cell. Whether it is the part of transverse colon, descending colon, rectum or anal canal, the number of the goblet cells are more. So we can easily differentiate, or keep one thing in your mind, between the small intestine and large intestine slide. The first thing is number of the goblet cells are more in the large intestine compared to the small intestine. No villi is seen in the large intestine. The villi are seen in the small intestine. So that is the basic difference. You will find villi, goblet cells. Say the slide is of small intestine, maybe the duodenum, maybe the jejunum, ileum. But no villi are seen, number of the goblet cells are more, the section is of large intestine. That is the basic difference. Now look this slide. The columnar epithelium you have seen, and villi, beside this, the epithelium is showing the projections, finger-like projections, just like the epithelium is showing the finger-like projections, and these finger-like projections are called the villi. So these finger-like projections are called the villi. And other important part, look here, between the columnar cells, you are finding the goblet cells. Now at this time, it should be clear in your mind how the goblet cell will be seen. Anyone? बोलो, जोर से बोलो, कोई बात नहीं, गलती करोगे, doesn't matter. गॉबलेट सेल कैसे दिखेंगे, ये भी है क्या? और नो, sit down. Goblet cells are looking empty. Look here, the goblet cells are looking empty. The nucleus is close to the basement membrane, flattened nucleus which is seen close to the basement membrane. दिख नहीं रहा आपको? ये देखिए, ये ट्रांसफर सेक्शन में देखिए. See, look in the transverse section. The nucleus is close to the basement membrane or not, and flattened. The answer of this, the goblet cells are filled up with the mucus, and that mucus is pushing down the nucleus. So nucleus is seen at the close to the basement membrane, and cells are looking empty. The answer is because the mucus, for seeing the mucus, we require the special stain. Then we can see that mucus is seen. Otherwise, in HNT stain, you cannot see the mucus. Because of that, the cell is looking empty. Okay. What type of this goblet cell? Unicellular or not? Okay. See the other important part. One thing, villi, and the invagination part is called the crypt. That is this one. This is the crypt, and the outward projection is called villi. And next to the muscularis mucosa, all right, everywhere we will find lamina propria lying here, filling the villi and rest of the part. And what are the structures seen in the lamina propria? Nerve fibers, blood vessels, and lymphatic tissue. Next to this, look this part. This is the submucosal part. Between the muscularis mucosa and muscle coat, what is that part? That is called submucosa. And submucosa is filled up with large number of the mucus secreting glands. When we see the submucosal part, it is filled up with the very much mucus secreting glands, and these mucus secreting glands are called Brunner's glands. Brunner's gland. So what is the Brunner's gland? Are the mucus secreting glands, and they are filled up the submucosa of the duodenum. So for identification of the slide of duodenum, between the columnar cells you will see the number of the goblet cells, but number of the goblet cells are not too much, they are less. We don't find any goblet cells in the stomach. Okay, the appearance of the goblet cells and the goblet cells number are less. The part of large number of the submucosal glands, that is mucus secreting glands, are filled up the submucosa, that is Brunner's gland. So by this, rest of the part is same, muscle coat, alright, same thing. And muscle coat is always, whatever the parts, is only the smooth muscle fibers. The smooth muscle fibers seen in all directions, longitudinal and transverse or circular. Adventitia is same, nothing important. So that is the section of duodenum. I will take the next class in the jejunum, ileum, appendix, colon, and all.